Good afternoon. Welcome to this edition of the Corbel webinar series. Today, we are very pleased to have with us Dr. Noriko Cassman from the Netherlands Cancer Institute to present Data Integration Platform for Cancer Research, cBioportal demo. My name is Marta Lloret, and I'm involved in the Corbel project on behalf of MBLBI. Before we start the webinar, I will remind you that this webinar is being recorded and it will be available on the Corbel YouTube channel and on our website. We have a Q&A session at the end of the webinar. And if you have any questions, please write them in the questions box on the GoToWebinar control panel. Before we start the webinar, I will briefly introduce you the Corbel project and today's speaker. Corbel is a Horizon 2020 funded project that brings together 13 research infrastructures from the field of biomedical science. And its aim is to transform the understanding of biological mechanisms and accelerate its translation into medical care. Modern biological and biomedical research involves complex projects and a variety of different technologies, and usually operate at the interface between different disciplines. Corbel will contribute to this project by harmonizing access and services for research involving more than one research infrastructure that offer biological and medical technologies, biological samples, and data services. Today's presenter is Noriko Cassman from the Netherlands Cancer Institute. She is bioinformatician. In 2008, she graduated with a BSc in biochemistry and psychology from Florida Tech. In 2013, after completing a master's thesis in viral metagenomics, she earned an MSc in bioinformatics and medical informatics from San Diego State University. There, she helped to teach a hands-on sequencing and genomics course featuring 454, Ion Torrent, and Illumina technologies. More recently, Noriko achieved her PhD from the University of Leiden in 2019 with research in soil microbial ecology conducted at the Netherlands Institute of Ecology. Later in 2019, she began working in the pathology department in the translational gastrointestinal oncology research group headed by Gerrit Meyer. As a part of the data team, she applies her genomics knowledge to the analysis and integration of oncological data to help build sustainable and robust research infrastructures for translational cancer research. At this point, I'll hand over to Noriko.
So hello and welcome everybody. Thanks for taking the time to tune into this webinar. Like Marta just mentioned, I'm a bioinformatician at the Netherlands Cancer Institute, and I work in the research group of Gerrit Meyer, and we do translational gastroenterology oncology. So today I'm here to tell you about cBioportal in my capacity as an operator for the National Health Research Infrastructure, HealthRI. So first, I will give some background on HealthRI. HealthRI is a partnership between more than 70 organizations in the Netherlands and Europe that are involved in healthcare and research. They bring together people from healthcare, academia, IT, and legal fields and work towards a national health data research infrastructure. So in Europe in 2018, there was a call for open science, which were a set of guidelines for the wide dissemination and accessibility of research publications and data. Implementing open science should ideally stream the process of translating science into better healthcare, which is the goal of research in groups like ours. So basically, we would want to make sure that the least number of people overall would suffer from cancer. But improving these translational research lines according to open science ideals, this can best happen when data is fair. So fair, I'm sure you've seen this paper, but FAIR stands for findable, accessible, interoperable, and reusable. And then finally, also here in the EU, patient data is also under the General Data Protection Regulations, the GDPR, and this safeguards patient information. So the challenge over the past 10 years has been to find IT solutions that fall under these different guidelines. To address these informatics challenges, the Translational IT or TRAIT project was launched in 2011 and spearheaded by our group leader, Gerrit Meijer. It's now one of the cornerstones of HealthRI and supports over 400 research projects, including about 4,000 individual researchers. So during a study, data can be captured in different ways. You see here the different domains that data can be captured throughout. So clinical data, biomedical imaging, pathology, biosample, and experimental data. This, of course, depends on your institute and whether you're doing a monocenter or multi-center study. But so you get data generated and captured. And these are some tools that we use and that are hosted by TRAIT. So the focus of TRAIT has been to host proven technology that could be adapted to specific needs of research projects, rather than embarking on large-scale software development projects. So in some cases like cBioportal, we are also involved in the development of the tool. So here at the bottom, you see the icon for cBioportal. This serves as a data integration platform in which the processed data from different data domains comes together for viewing, query, and analysis. This is the platform that I'm going to be demoing today. So we have to remember that after spending lots of time, effort, and money on a research project, the work is still not done unless the data is in a shareable format. So we're asking the question, what is a good way to make my data immortal, or as immortal as possible? So in our group, we choose to use cBioportal. And so here's the public cBioportal. It's at www.cbioportal.org. This allows us to integrate different types of data, as well as to store, view, analyze, and access these valuable data sets. So here you see a screenshot of its user-friendly front-end interface. Down here at the bottom right, they keep a tally of how many studies are in there. You can see that there's about 281 right now across different types of cancers. So this public cBioportal is hosted in New York at Memorial Sloan Kettering in New York. Let's see, this public software is open source and available in GitHub, and there's a very dedicated developer community. So this public cBioportal also has a lot of resources like tutorials and step-by-step guides for further reference, which are all available under this tab, the tutorials tab. So as a repository, the public cBioportal also has all these public studies available. You can just go to the datasets tab here, and then you see all the studies that are on there and how to download it using this download button. So that's the public cBioportal. And then for European cancer studies, specifically Dutch ones, HealthRI offers a national solution of cBioportal. So if you go to cBioportal.health-ri.nl, you'll see this login page. If you want to get access to the national cBioportal, you can contact the HealthRI service desk or follow the email link as you see here on the login page. Okay, and here is a screenshot of me having logged in. So you can see these are the studies that I have access to. And again, this is a national cBioportal, so it's hosted by HealthRI. So this instance is especially secure, and it has servers located in the Netherlands. The data on the national cBioportal is private, and you control access to your own studies. You can easily share your data with selected collaborators. And finally, you can compare across studies, given a standardized vocabulary between studies. So when we go to use cBioportal, and shortly I will switch to the actual cBioportal instead of a screenshot, and hopefully the connection stays good. But if I were to select something, I would select, today I'm selecting this public study at the bottom, and I would click the button Explore Selected Studies. And let's see, switching to the cBioportal. Okay, so then you would see the study view. So here we are in cBioportal. You can see the URL at the top. And let me just switch to this for just 1 second. Yes. Okay, so this is the public study that we're looking at today. This is the low-grade gliomas public data set, published in Science in 2014, associated with this publication by Johnson et al. So they basically compared mutations in recurrent and primary tumors using whole exome sequencing. And now we're going to take a look at this study as it is in cBioportal. Okay, so like I mentioned, this is the public study view. Here you can see all the clinical data. It's in a user-friendly view. So all this clinical data gets summarized in pie charts or histograms here. You can see all the different types of data that was uploaded for this project. Let's see. Have to move that. Okay. Okay, so in this front dashboard, you can remove charts as you want, or you can also reselect them here and see different variables that you would like to view. So that's all here. If you want to look at the clinical data in a tabular format, you can go over here to click the clinical data tab. And you can see all the variables here listed. And what's especially nice in cBioportal is that you have patient information as well as sample information. And the way it's organized is you can have multiple samples per patient. So if we go further here and look at the information for one patient. So this is the clinical data view for one patient. You can see that information here. So these are some attributes, extra attributes so you can add on. Yeah, so up here we have the timeline data for this patient. Different subsections like when the surgery happened, treatment, specific treatment. So all that goes on the timeline. Down here we also have the map of the genome with the different chromosomes, and then the location of the different mutations. And then also down here in this mutation section, there's also more information about the genes, the specific mutation, and other metadata, other information. So like I mentioned, there's multiple samples per patient. This is all integrated together so that this first sample for this patient from the primary tumor is here, and these are the mutations associated with it on the top. yeah, so this one circle corresponds to that first sample. And then here, the second circle corresponds to that second sample. So this is all integrated. You can already see that for this patient, there's a lot more mutations than for the first sample. So the recurrent sample for this patient had a higher mutation count. Okay, and then if you go back to the clinical data for this one patient, then you can see that all that information stays here and is accessible. Okay, so this is, again, going back to the study view. Again, I'm here selecting the study, then exploring it. And here at the front end first dashboard. So yeah, going back to the variables, you can also make sub-selections of variables that you want to see. So if you wanted to select only those patients with a certain subtype of cancer, then you can focus on those. Yeah, so it's a pretty user-friendly and easy to understand interface. And what's especially nice about it is that here you can always download any of this data at any time. You just click on these download icons, and then you can save the file to your computer and look at the data that way. And also use it for further analyses using R or something more complex. Okay, so this is basically the study view. You can also go back to the start page and then do the query by gene. So this looks at the data, the study data based on gene. So I've already done this for a list of genes. But basically, you enter the genes you want to see here, and then you submit your query. So I did that already. So I just put in these 6 gene IDs in there. And then you get to this gene-centric view. So it shows oncoprints of the different genes. You can also add clinical data to this view. So here I'm going to go ahead and add, let's add sample type, mutation count, overall survival, and take a look at those. And you can see here it's added these data to your view. So what if we view events per sample, and then because in the study they were looking at primary versus recurrent, so let's do that too. Move this up and then sort it. So you can see it organized the data with based on the sample type. So here are the primary samples, and then here are the recurrent samples. And then looking at this, you can see that the mutation count, you can see that basically in a few of the samples, it looks like there's more mutations in the recurrent sample types compared to the primary. So that goes with the goals of the paper. Okay, so if we go to the plots tab, you can also look into this data a bit more. So let's do, yeah, clinical attribute on the horizontal axis. What do we want there? We'll put sample type there. And then on the vertical axis, we'll put non-silent mutations in these genes. Okay, so here you get a plot, and these are the non-silent mutations in TP53, ATRX, CIC, FUBP1. These are the numbers of samples that they're in. And here on the x-axis is the type of sample. This is all from the clinical data, so you get the primary and recurrent samples. And so what I see right away from this graph is that, so there's a mutation in CIC that's only in the recurrent samples. And when I mouse over, it looks like that's in one sample there. Whereas it looks like both the primary and the recurrent samples have this mutation in ATRX. They also have both TP53 and ATRX mutations. But this TP53, it looks like there's more samples with mutations in this gene compared to this gene. And yep, this is 3 samples here, and then 5 samples have that here. So let's look a bit more at that. Let's go to the mutation tab. Okay, so here we can look at TP53, that gene that had a lot more samples with mutations in this gene in the recurrent samples. And you can see here, maybe first I'll look at a different one. Oh, no, maybe not. Maybe I'll go back to that one. Okay, so here, yeah. So here was the one with a lot of mutations in it. And then if you mouse over here, this is the most common mutation. There were lots of samples with this here. You can see the specific amino acid change. And if you select it, then you can get all that information from these here. So what patients they were in, what cancer type they're from, the protein change, also all the clinical data. Okay. Okay, and then this was one of the genes that didn't have very many differences between the primary and recurrent. So, but you do see them in both. So here you see that too. So, okay, so I'm showing just a few tabs, but there's also these different tabs here. And go back to the plot. So this is also something you can see that goes along with, I think, figure 3 in their publication. This is also basically what they were saying in that figure as well. Okay, and then one last thing I want to show with this section, the gene-centric place, is this download tab. So all of this data can also be downloaded. Again, anyone who is has access to the study that you control, they can download it here. Yeah, okay, so with that, I'm going to go back to the presentation. Let me find my place. Okay, so I just showed you cBioportal, and here are the people at the NKI who are also involved with uploading studies to cBioportal. Here are my colleagues, Brenda, Jan, and I. So now I'll talk a little bit more about what we do behind the scenes. So how does a study get put into cBioportal? What happens to the data? How does that have to look like? So what we receive is, of course, a study data, the data of a study. So basically, we receive formatted files from researchers or from a research group, and then we upload these to cBioportal. This is an example of what we would get and what we could upload. So these are all tab delimited text files. Just in a directory. So the main thing to notice is that there's a bunch of files called data files, and then metadata files. And these metadata files refer to these data files. So this list is also something you can get if you download the datasets from the cBioportal. So you can take a look at how they're organized as well if you want to. So here's an example of the clinical data file of de-identified patient information and the meta file that refers to the data file. So up here is data clinical patient, and it's got a particular format. It starts with 4 header lines describing the header information. So the name is something that you would see on the front end or on the user interface that you see through the website. And then in the back end or the database behind cBioportal, it's organized with these headers after these first 4 lines, if you can see that. Okay, and so that's the data file, and then this meta file at the bottom is something that refers to the data file. So it just explicitly says the file name, and that helps keep everything organized in the back end. So that was the data, one of the clinical data files. So here's an example of the one of the timeline files. So this is different from the clinical data file in that it doesn't have those first 4 headers. It just has the database headers and then the data. And then the meta file refers to the data file here explicitly, as you can see. Okay, so again, the format and controlled vocabulary in these behind the scenes files does ensure what we see on the front end website that the users can access. So here I showed the clinical data, but cBioportal, of course, was also designed for multi-dimensional genomics data. So it has the capacity to include other data sets, for example, copy number, expression, fusion, methylation, and protein level data. And if you go to those public data sets, you can also get an idea of what those should look like if you want to put your data into cBioportal. But cBioportal also has this handy guide for what these files to be uploaded should look like for all the different data types. These are also very handy if you want to prepare your data management plan before your study is going to be executed. So I will be linking this page and all the links at the end of this presentation. So at HealthRI, we also do provide advice on how to format your files and whether your data type is supported and other questions like that. We do not currently provide like the actual formatting. That would be something that a data manager or bioinformatician in the research group would do. So to conclude, I would say we have jumped onto the cBioportal bandwagon as a group and also at HealthRI, and are very happy with the lively development community surrounding cBioportal, both the international and the national. We're happy with the functionalities and sharing capabilities of it, especially the ability to download whatever view you want to see. And so some of our tips for success with using cBioportal are to first start with a data management plan per project. This includes distinguishing between, for example, internal data and data that will be shared. To include an overview of the data flows between data sources and where it will be deposited, or the repositories. To try to keep your data as clean as possible. This includes agreeing on variable names beforehand, using official guidelines where possible, keeping data as free of noise as possible, and also finally curating your data before going through the formatting steps. Okay, so with that, I will leave you with some further resources. Here are links to the Health RI website, the service desk, where you can register to use the National cBioportal and also get more information. Here are links to the public and national cBioportals. For those of you who want to explore cBioportal further, here's the link to the formatting guidelines and our step-by-step user demo as a PDF. So with that, I would like to open the webinar to a question and answer session. And for this, my colleague Mariska Bierkens at the NKI will also join me to answer some of the user questions. So thanks for your attention.
So yeah, Noriko, we already have some questions that people send in their registrations, and we have a couple already in the question box, but please, if anyone has other questions, please just add them to the question box in GoToWebinar. So I'll ask with one of the ones we received previously, is how can this portal be used during a clinical trial?
So Nori, shall I answer that one?
Sure, Mariska.
Hi, so hello everyone. I'm Mariska Bierkens. So how can this portal be used during a clinical trial? Well, as Nori indicated during the presentation, cBioportal is a data integration platform where all data comes together from different domain fields. And it is not a clinical decision support system, but really a platform meant for integrating data and seeing what data has been gathered already, bringing it all together and making it low threshold for the researchers performing, for instance, then a clinical trial to see the status of an up and running system. So yeah, it's really meant for exploring, reviewing, and querying the available data that you have as a complementary platform for your other clinical trial systems. So I hope that answered the question of the one that sent it. If not, please use the comment section to ask clarification and can add more.
Thank you, Mariska. So we have a couple questions that go in the same direction. So I'll ask you all together, and then you choose how you answer this. So it would be, what are other platforms similar to cBioportal currently available, and how is cBioportal different? And then the other similar question is, what makes Health RI better than TGCA, Cosmic, or other cancer data resources?
Starting what platforms are out there? Well, that is a difficult question to answer. Like you saw in the Trade App Store that there was a different logo there called Transmart, which is more for subject level querying of your data. But that doesn't focus as much on genetic data. And there are different commercial companies that are offering to build customized solutions to solve your data integration needs. So yes, there are different platforms out there. And cBioportal, as Nori mentioned, it is an open source platform. It has a vibrant community in which researchers together with technical developers are working together to see to their needs. It's possible to ask for development, and then the processes are aligned in such a way that it is useful to the user. And what content-wise, cBioportal generalizes the data. It is gene-centric, and you can view your data patient level, sample level, gene-centric level, and one feature that was not shown explicitly, but there are group comparison levels possible in cBioportal as well. And because it simplifies the data to a gene-centric level, it makes it much easier to do all these integrative analysis to look at what is the status of the TP53 mutated patients versus not, and to view your data in such a manner without consulting a bioinformatician. And there are also all these little hover over functionalities, rich data formatting in place that can link out to FDA approved drugs, and then different levels indicating whether a drug is available, OncoKB and other sources. So it is linking out to other systems with knowledges in such a sense. So there are also other platforms like Molgenis, for instance, and Molgenis experience tells us is more for a bioinformatic user. It is difficult to get the data in, but it is completely customizable then by the people that wish to use such a platform. But if you then are like, okay, can I, who am running a clinical trial, easily access my data and do some integrative analysis? The answer is no then. But if you are a bioinformatician and you want to know, okay, I have a VCF file and I want to know if there is an A to G, blah, 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 then the bioinformatician could retrieve an answer on the different platform. So I would like to keep the answer to that one, if it's all right.
That's okay. Thank you, Mariska. We also have a question about how can you access the data from the command line?
Nori, you would know that. Noriko?
Yes, Marta, sorry, can you repeat the question?
Yeah, it's how can you access the data from the command line?
Well, like I showed the website, so that's basically the front end of the like the user interface for the data. If you wanted to access it on the command line, well, one, you could access the backend data, those flat text files that I showed. So I guess
okay, so
what I understand is you want to be able to access your data on the command line, but I guess if you want to do that, you already could, because before you can access it on the front end through the website, you have to upload it as those text files. So if you do that, then you already have access to those. Does that make sense?
So, and if I might add to that,
sure.
If people are interested in the source data underlying cBioportal, indeed you can download it, the process data using the tabs as Nori showed. On the other hand, if you are more a domain expert in the bioinformatic field interested in the raw data, then in cBioportal, the data owners have the possibility of entering metadata, linking to raw data repositories. Nori showed those for the TCGA. There's also the European Genome Archive, in which users have to go through a data access committee in order to obtain the data. Those are also possible ways for having a look at the available data.
There is no command line interface for cBioportal, but you can always download any of the data that you upload or have it available from what you upload.
Okay, thank you. Another question is, can it perform advanced statistics? Because I can see RMATLAB.
So I guess that depends on what you mean by advanced, but there are some different statistical tests that you can use in the gene-centric view. It does t-tests and that sort of thing, group tests, group comparisons. Again, I don't know what specific analysis you're looking for, but it has a lot of like viewing and basic analysis capabilities, I would say. But there's a lot built into it already, and there's also the possibility for asking for that particular analysis that you're interested in, and then having that as a new feature available.
Okay, thank you, Nori. Then we have some more questions here. One of them is, I was wondering that if researchers are working on different aspects of breast cancer, can we use it to integrate their findings?
Okay, so I'll answer that one, Nori. So if you have these different data sets that you wish to combine, then the short answer is yes. And that is possible. You just need to make sure that there is approval from the sites to which the data belongs. So you need to gather the data. Then you need to harmonize the data to make sure that you are reporting the data in the same fashion. If, for instance, very simple example, if you are having a study with female and male and want to compare it with woman and men, the data items won't match automatically for those data attributes, and then you can't combine the data. But if you say, this is the codebook that I'm going to use, this is the terminologies, then we're combining the data. Then there's the functionality of either uploading the separate data sources as a separate study and then combining it, which could, for instance, be done if you have different ongoing studies. Or you could just combine all the study data into one larger study and then view all the data together.
Okay,
So,
thank you. Can you please clarify whether the platform contains gene expression, methylation, or imaging data?
Nori, I'll leave that one to you.
Methylation, yes. What are the other ones, Marta?
Gene expression or imaging data?
Yes, gene expression also. That was one of the data types that I mentioned in the file format, so that is supported. And then imaging, also, yes. You can also put in digital pathology slides and that kind of thing. Hopefully that's the type of imaging that you're talking about. Yeah, but if you want to look at all the data types that are supported, I would suggest looking at that file format page. But it does have what the majority of data types that we would need, actually.
Okay, and is there a way to only select the studies based on features like RNA-seq to compare only this?
Is there a way to, there is a way to search for studies. I'm not sure specifically based on what they have, but there is a search box in that first overview when you log into the portal, when you log into the cBioportal. So I guess I've never tried that, but I think you can.
So yes, you definitely can. If there is a tab called datasets in cBioportal, and if you click on it, you can already see CNA or RNA-seq. You can see different data types of omics data to give an indication of which studies have done what type of omics profiling.
Okay, thank you. Another question is whether the only way to access the platform's metadata is by asking for permission.
So, okay, there are two different platforms. One is the public version standing in the United States of America. And you don't need a login here. These are public studies, mostly TCGA studies. And if you are working on studies and you are having a partner in the Netherlands, then you can make use of the Health to Right platform. And then you do need the login because the GDPR is all about patient privacy and security. And unless a patient or subject has given their explicit consent that their public can be made publicly available to everyone, we need to ensure that there are correct data access procedures in place. And we can only do that if the studies are private. And that's why the way is, okay, contact the service desk, upload a study, and then also indicate who is the contact person for the study. And only after approval by the institutional review boards or other procedures may another person have access if the data is still private, actually, which is something that we'll be seeing more and more in the future.
Okay, thank you, Mariska. Does the platform contain also health data records from the patients?
The electronic health records, they are, well, can be uploaded, and it depends on the study which data attributes are being collected. So far, not all data is being copied and imported into cBioportal. Some attributes are left out that contain patient privacy sensitive data. But if you have clinical data attributes or sample data attributes, Nori showed a big tabular file. Yes, if you supply it in, then it can be imported and it will be present in cBioportal.
Thank you very much, everybody, for attending this webinar. Thank you, Nori and Mariska, for helping us with understanding cBioportal. Then I would like to let you know that the next webinar of the Corbel webinar series will be on the 21st of April, and it's about the BBMRI LC services across the nodes of biomedical sciences research infrastructures. And it will be Michaela Meyerhofer from BBMRI ERIC who will be presenting it. And if you want to know more about it or register, please visit our website.